Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog. Good evening, I'm Yvonne Stapp for Science for the Public, and we're pleased to join LinkedIn tonight at Robbins Library for a special presentation on viruses by a distinguished expert, Dr. John Connor. Dr. Connor is an associate professor of microbiology at the Boston University School of Medicine. His lab is well known for innovations in virus research and also in new technologies for virus identification. You can visit the page for the event on our website to get articles and other information about his work. He's been in the news quite a bit. Dr. Connor received his PhD from Duke University and joined the faculty of Boston University in 2007. His background represents an important trend in science today and certainly in virus research. The convergence of fields, in his case, expertise in pharmacology, genetics, molecular biology, and engineering, quite a lot. A major area of Dr. Connor's research focus is on the mechanisms by which viruses take over the cell's machinery to replicate themselves. An understanding of this process is essential to the development of inoculations against infection. Another area of Dr. Connor's research involves the development of point-of-care virus identification tests. He'll explain the challenges of this process. He'll also explain the great difficulties of containing viral infections in today's global community, where travel and urbanization facilitate the spread of viruses. Tonight, we'll leave here with a healthy respect for viruses and a great appreciation for the work of scientists like Dr. Connor, who are contributing so much to public understanding of viruses on the one hand, and to the medical advancements that will prevent infections on the other. We're very honored to welcome Dr. John Connor.
Thanks, Yvonne. I think I'm talking in a way that folks can hear me. If I begin to trail off, somebody please just let me know. We've seen what I'm guessing will be my first slide. I thought I would start just by introducing who I am and the things that motivate me. I'm a microbiologist, which means I study small things all day. This is me studying for my first exam. Most relevant to this talk, most of what I do is study viruses and how they function. In terms of where I do that work here. This is the National Emerging Infectious Diseases Laboratories. It is just adjacent to Boston Medical Center, part of Boston University School of Medicine. This beautiful blue sky picture, I don't know how long it took them to find this day, but it wasn't this day that they took that picture. I thought I'd start out by really defining the bacteria and viruses, the differences between them, and focus then in on my particular interest, which is viruses. The field of microbiology studies fungi and bacteria and viruses, all of which are really interesting, but one person can't look at all of them. And my focus on viruses has been driven in part by the differences between bacteria and the interest that I find in understanding how viruses function. So from my perspective, the way I look at bacteria is that they are able to replicate and divide on their own. They can live in a bunch of different environments, and they have thousands of genes. They're genetically fairly complex. And this is of stark contrast to viruses, which are obligate parasites. They can't make copies of themselves without any help. And that means they have to find a host. And they essentially have to figure out how to take advantage of that host and how, in fact, to steal a lot of things from the host. In terms of comparing them to bacteria, viruses are genetically very simple. They can have anywhere from 1 to at most 300 genes. The overwhelming majority of viruses, you'll find that they have about a dozen, maybe 2 dozen proteins. Again, they're obligate parasites, and they're incredibly small. And just to give you an idea of how small they are, my laboratory can't use a regular microscope to look at them. However, we still think they're very captivating. What I'm putting up here is just a general size diagram ranging from below a nanometer to about a millimeter. And what you can see is that at the low end of this scale, you can see atoms. At the upper end of the scale is a frog egg. So at no point are we talking large things. But when you ask where viruses land within this, they're right in this range, which is viruses range in size somewhere around 100 nanometers. In terms of being able to look at them, light microscopy will allow you to see bacteria, it'll allow you to see mitochondria, as well as cells of all different types. It fails to allow you to see flu viruses or any virus of that type. You have to use much larger, much more expensive microscopes, such as electron microscopes. However, doing this allows you to see a lot of pretty neat things. And this is just one of those things. So what I'm showing you here is an electron micrograph of a cell that was infected with a virus. And then a little bit after infection, people looked at what was happening right at the edge of the cell. So what you can see here, this dark mass is essentially a lot of cellular protein. There's a cell membrane right here. And these things right here popping off are viruses that are budding from the cell. So the virus has used the cell to its own advantage. There's a lot of viral proteins. There's viral nucleic acid poised right here. And you can essentially see the process of new viruses being budded off and then floating away from the cell. You can begin to see in this floating away from the cell, it looks like these budded virions actually have a significant amount of structure to them. And when you take viruses after they've gone through this budding process and look at them in the electron micrograph, you see exactly that. Right, so this is just a collection of viruses that have been purified. You can see they're all the same shape, they're all the same size. And you can, within that image, begin to see a glimmer of additional structure that must be present based on the repeating patterns you see in light and shadow there. And if you pile a lot of these electron micrographs up, you can begin to get a very good idea of what viruses look like. You can identify their three-dimensional shape. Even though we have a hard time doing it by conventional microscopy, electron microscopy really allows you to see what's going on. And this is just an example of one virus. This happens to be dengue, which is the causative agent of dengue fever. Dengue is a virus that uses proteins to make up its outer shell. You can see them in various different colors here. You can see how they are put together. You can see this five-fold symmetry here. It looks like a sea star, except with a little hole in it. This is where signals from the outside can go in, and also where nucleic acid that's stuck inside can eventually come out. You can see the other colors representing different proteins with different types of symmetry. This is a two-fold symmetry. Here you have three-fold symmetry. And all these fit together very nicely to make an elegant casing for the virus's genetic material, which is going to carry from one place to the next. And this type of shape is very common in viruses. However, if I can go the right direction, it's not the only shape. So this is another image I'm showing you here, adenovirus. You can see it has that same central, almost buckyball-like shape. However, this virus manages to put spikes on the end that help it attach to the cells that they want to enter and get in. This shape is not only interesting from my point of view as a scientist, you also find people that are captivated it for other reasons. I grabbed this image from an artist who's decided that looking at electron micrographs and cartoons wasn't nearly the right way to look at viruses. He actually blows glass, and this is a coronavirus that he put together out of a number of different glass components. You can see the outside of the virus, and the nice part about the glass image is you can see straight through into the genetic material. And I just put these together because I think they're really pretty. This is just another way of lighting that same type of structure. So viruses are really small. They have a very nice, very tight architecture, and they're fun to stare at. They're fun to do art with. But on the other hand, viruses are incredibly dangerous. Viruses are one of the greatest threats to human health that have been seen throughout human history. Smallpox is estimated to have killed at least 300 million people. This is the 20th century estimate alone. There are probably many more before that. Influenza, I have no doubt have gotten lots of talks about how you should get your flu shot, and you should. But it is a disease that continues to afflict humanity. The 1918 flu resulted in at least 100 million deaths. HIV is another example of a virus that can take hold of the human population, going from a non-existent virus to a very well-established virus now that has a very large impact on human health. These are what I would consider known threats, but what I also want to point out is they're not the only ones. I just sat down and in a few minutes came up with a large additional set of viruses that are emerging, where people understand that these viruses exist. They've caused human disease, in some cases very severe human disease, but in most cases they have not really become what would be considered an epidemic, which is a large number of cases. What they are very good at is causing a lot of fear. Here, what you can see is the response to the SARS outbreak in 2003. This is just a group of people who are trying to prevent transmission by wearing ear loop masks. This is an example of an Ebola response. You can see it's a little more severe than what's going on here. The individuals are going out into a resource-poor area, fully clothed, in order to try to keep any skin-to-skin contact, any contact with infected fluid from occurring in situations where they feel that Ebola might be present. One of the things I want to emphasize by both of the things I've told you is that I think viruses are a paradox. They're both very simple and they're dangerous and complex at the same time. The fact that they're small and genetically simple makes me think of them as a toy boat. They, however, have quite an ability to cause harm to humans, which are much more genetically complex. Right? We've got about 25,000 different proteins. So in this analogy, we appear to be the Nimitz-class aircraft carrier, and the virus is the toy boat. However, in terms of who ends up winning an encounter, often it's the virus. Often it's the toy boat that seems to do a much better job of causing harm. And it does so through a fairly set pattern. Right? All the viruses that I've mentioned essentially do the same basic thing from a 30,000 foot view. Right? A virus will come and attach to a cell. It will then insert its genetic material into the cell along with some support machinery and use that cell that it infects in order to make copies of itself. Once it's made a lot of copies of its genetic material, it then packages that genetic material to make new virions, usually hundreds or thousands from an individual cell, and goes and starts that cycle again. If you think about that in terms of a multicellular event, so these are a bunch of different cells. They're not olives, though I recognize the similarity. If one virus comes in and infects one of these cells, makes copies of itself, it then spreads it to the neighboring cells, and this happens over and over and over. And this is what takes virus infection and changes it to virus consequence. And there are many consequences, from sneezing to rashes to downright acute feeling badness, and in some cases very severe disease that can lead to hospitalization. And as I illustrated on the earlier slide, viruses really can be associated with a variety of events. either sniffles and feeling cruddy, that's usually rhinoviruses, several different adenoviruses, but they can also be associated with high fatality. And one of the things that has been a very serious question is, how do you know what virus is going to do what in terms of causing significant illness? So because there are so many things that are unknown, I have things to do during the day and on the weekends. And a lot of the things are trying to answer the questions that are up here, right? Trying to get past the fact that right now we don't know as much about viruses as we want to. And I think that puts us at a disadvantage in what is an ongoing competition. And the things we are really sort of unclear on is, how is it sort of mechanistically that they make us sick? What is it that makes us get either just a weak cold or a very strong life-threatening disease? And also, equally importantly, how do we detect viruses quickly? In all cases where you get a virus, if you need to be treated, the faster you can identify that infection, the more likely you are to have a happy prognosis if you're caught early. And one of the other things that my lab spends some time on is understanding how you can limit disease. How do you develop new treatments and vaccines? And the idea behind this is that my lab, other labs are working to make certain that we're prepared against both existing and emerging viruses. And as I put up earlier, there's lots of examples of this, including SARS, the recently identified Middle Eastern respiratory virus, chikungunya, which is steadily hopping its way across the globe to be closer to being endemic in the United States. And there are also other viruses that I would say drive this home directly. And currently, the object lesson and why we need to know more is Ebola virus. So this is an electron micrograph showing Ebola. It's not a round virus, which is what I'd showed you earlier. It is what's called a filovirus. You can see its nice extended sort of rope-like shape. And you may have noticed Ebola has been in the news a little bit recently. What I think about when I think of the virus are the points here, and that is that Ebola virus has been known since 1976 to be a very severe virus that causes what's called hemorrhagic fever. And it's essentially the paradigm pathogen of this type of disease. Infection with the virus leads to a very severe infection that is associated at end stages with uncontrolled bleeding, which is how you get the moniker of being a hemorrhagic fever. In terms of what type of virus it is, it's called a filovirus because it has that filamentous shape that you saw on the previous slide. In terms of the details, it's a non-segmented negative strand RNA virus. I would love to talk about that for the rest of the time, but I'll save you guys the pain of it. One of the real important aspects here is right now there's no specific treatment or vaccine for Ebola. This was brought to the fore. It has been known previously that this was going to be an issue. There have been at least 20 outbreaks of Ebola and also Marburg, which is a cousin virus of Ebola in Africa over the last number of years. And every time these outbreaks happened, the comment was made, there really is no treatment. We really need a treatment. And every time the outbreaks were fairly small. Right now, the ongoing outbreak has completely changed that calculus. This is just an image here of all the different outbreaks that are known to have occurred. You can see most of them cluster in Central Africa right here. Up until this spring, it was thought that there was no Ebola virus in Western Africa. However, as we know now, that's not true. In terms of how Ebola virus jumps into the human population, as I said, it's been responsible for sporadic outbreaks. And people have been trying to understand how that is, because if it was something like smallpox, which is only transmitted by humans, you wouldn't get sporadic outbreaks. You would get a constant set of infections. What's believed to happen now is that the virus exists in a reservoir, meaning some other animal in which it's able to replicate happily and not cause severe disease. The most popular hypothesis is that this reservoir is bats. And that these bats, through still an unknown mechanism, transmit it to humans. It's not very easy because it doesn't happen very frequently, these outbreaks. But it's known that there are some antelope and also that non-human primates can support this virus, that can replicate the virus. And it's known in the places where outbreaks occur, people end up eating all three of these things. And so it's likely that part of the jump is made when humans come into very close contact with an infected animal. Maybe an animal that was especially easy to catch because it was sick. It's not really known, but it is direct contact and requires direct contact for transmission. So I have sort of bled into talking about this virus because there is an outbreak in Western Africa now. And the way that this outbreak came about, I think, is a perfect example, just generally, of how virus infection can bloom and how it can go from being something that very few people think about to being something that is a very serious concern to the world. Prior to 2014, Ebola was unknown in Western Africa, and that led to a sense of complacency. And today, that has dramatically changed. If you look at the current outbreak and what has happened, it was recognized that there was an outbreak in March, and the number of cases I'm showing you here, this is the number of new cases tracked over time. It's split up by country, where Guinea is in blue, Liberia is in green, Sierra Leone is this maroon color. Nigeria, for those of you who have really sharp eyes, is stuck right in here in gray. So what you can see is that this outbreak happened as one would expect a classic outbreak to happen. There were a few initial cases. Those initial cases spread it to others. You can see that in Guinea, what happened is that the outbreak sparked. You got an increase in cases. It then looked like it was resolving until there was some sort of additional transmission event. That transmission event spilled from Guinea into Liberia and into Sierra Leone. And at that point, the outbreak has cascaded and increased weekly. Recently, there's been suggestion that it has not been expanding, but that's the first indication over the last 2 weeks that it was not continually expanding. This is data that only goes up to September. I thought I would also put in the graph here, which I think is fairly informative as well. I took this from The Economist, which has been doing a very good job of trying to visualize what's going on. So this is showing Western Africa, where the major outbreak is happening. This is showing the number of infections in the large circle, the number of deaths in the dark circle that's slightly smaller. What you can see is the infections that are here in Guinea, Sierra Leone, and Liberia. You can also see right over here these cases in the Congo. It turns out that there was a separate outbreak of Ebola that also happened, that began in this summer and was successfully contained and closed down. The country was officially declared Ebola-free a month ago. But this just illustrates that these types of outbreaks are happening, and they're happening not regularly, but with increasing frequency if you look over the last 20 years. This outbreak has led to spillover not only to Senegal and Mali, which are right next door, but also to Nigeria through plane travel. When you look at the severity of this outbreak, what you can see is this is when they first discovered Ebola. There were a couple hundred cases in this outbreak. You can see a huge space where there was no more introduction, and then as we get into the 90s and early 2000s, you can see that there begin to be more and more regular outbreaks. This single outbreak here has been catastrophic when you compare it to these other outbreaks in terms of the number of people infected. And again, that's the light bar, and the number of people that have succumbed to disease, which is in the dark bar. So this has really changed how people understand the threat that Ebola faces, but this is something that has been seen before in terms of what do viruses do in an outbreak. The 1918 flu would show a much more dramatic graph. The and similar analysis of the HIV epidemic would show a much more dramatic graph, but still this is very telling in terms of what can happen to an infectious disease when you have an outbreak scenario. And of course, one of the things that is more and more recognizable is when something happens anywhere in the world right now, it is not necessarily that far from anybody's door. What I put up here is just a map of air travel. This is, in the green traces are individual flights leaving from one country and going to another. And what you can see here is that pretty much all of the continents are well connected in some way or another. You can go from Africa to Europe to anywhere. You can go from South America to either Europe or the United States, and then anywhere. And what this means is that when there is an infection, people can get on a plane and people can move quickly. We've seen that. That happened, and it happened because when you start looking in detail at that air flight map, what you can see is from Western Africa, there are regular flights of people going to the US, going to Europe, going to Asia, even going to other parts of Africa. So this type of travel means any type of infection event that happens here, or really anywhere, can start moving. You can imagine this same thing being looked at in a reverse line where people can say, hey, those things that happen over in North America might be moved to us, and in fact might have been moved to us. And so now we're in a situation where, as one might predict, air travel has moved the virus. Air travel has moved the virus from Western Africa to the United States. It's moved it to Spain, where there have been either unintentional infectious events or transmission events when people that were known to have the disease were brought back to the home country. I'm hopeful that this number of countries won't get any larger, but I also think that it is a serious thing to consider. One of the things that happens when these outbreaks happen is that you have a second dependent outbreak, and that is an outbreak of concern and fear and hyperbole. And here I just went and pulled a couple of points here. You can see that the major news outlets are really trying to understand what to do here and trying to compare it to other current events, or trying to understand whether the country should take severe measures in order to deal with the fear that's associated with an outbreak of a virus. In this case, it's Ebola, but it could be any virus. And one of the things that's especially apparent is when these outbreaks happen, all of a sudden people want answers to very specific questions, and they're here. The questions are, where are the treatments? And where are the vaccines? And where are the diagnostics? And why are they not on the shelf? Because now we need them. And the underlying question is, why hasn't anybody been thinking about this, that we got hit with it? And one of the reasons I'm here is to say, actually, people have been thinking about this. Before this particular outbreak, there have been dozens of scientists that had already been concerned about this and working on it. And significant progress has been made. And that is in large part thanks to the American taxpayer. So you guys should be proud of that. One of the things I wanted to put up is a comparison of the SARS outbreak and the Ebola outbreak, the current Ebola outbreak. When SARS was first discovered and people started asking these general questions, one of the first things they wanted to know is there a diagnostic test for the virus? And at the beginning, the answer was no. They wanted to know, is there any treatment? And the answer was no, much less one that was FDA approved. And the second well, I guess the fourth thing they wanted to know was, how's it transmitted? And at the beginning, it was really an open question. It really was not known. There were a lot of unknowns and a lot of things that people had to find out fairly quickly. In terms of this outbreak, a lot of these questions can be answered in the affirmative. When Ebola was diagnosed in Western Africa, the first tests, first diagnostic tests got there within a week. They have been improved since then, but in terms of response time, it was incredibly fast. There are several treatments that are already in advanced testing for this virus. So when people say, where is the vaccine? The answer actually is there are 3 good candidates. And all of them are now in phase 1, making certain that they're safe before they get rolled out. And a great deal actually is known about how this virus is transmitted and how this virus gets moved from one person to another, which is another potential advantage in terms of being able to address the outbreak and being able to address what's going on. So I say that to say that in these situations, there's a great deal that needs to be known, but also that through a lot of investment, it is possible to have most of that ready to use. Now, I also want to say there's a lot more that still needs to be accomplished. And one of the things that I'll highlight is it would be very nice to have an FDA-approved vaccine for Ebola. That was a statement that a lot of people made in 2011, and it didn't happen because of a lack of financial push. But I think it will happen now. It would also be really nice to have drugs that can alleviate suffering from the disease. Again, there are a lot of people that are working on that. There are some advanced stage compounds that could be very effective. And one of the things that I think is really still the unsolved problem that is plaguing the current outbreak is the ability to do easy diagnosis. It's something that is easy to say, but it is hard to do. These are resource-limiting environments where it's very hard to get power, much less a good diagnostic test to where you want to go. So having said all that as an introduction, I'm of course going to talk about some of the work that my lab does that directly tries to address this. And I'm fortunate to work with a lot of people who have the same goal, which is we would like to try and provide solutions to the existing challenges for infectious disease. And we have a number of different things that we look at. One of the things that we're interested in is trying to find small molecules that will derail virus replication. We've been interested in Ebola for a long time, and so we've been looking at that. And generally what the question is can we find a small molecule, something that is not necessarily a drug, but that is a probe, a lead that can identify what kind of drug you'd want to make that will stop the virus from making copies of itself. And essentially how we do this is we've worked in lab to set up a bunch of tests that can follow virus replication. And we have wonderful collaborators in the chemistry department at Boston University who have a separate question, which is how can we make the neatest and most complicated molecules that have drug-like properties? And we work together. And essentially what we do is my interest is in running these compounds through these tests to try to find ones that keep viruses from working. So we throw a whole bunch of compounds in to these tests, and what we want to know is what comes out. When we run the tests well, we'll find something, a molecule that looks like this, for instance. And it allows us to then test it and say, okay, this worked in our specific test in the lab. Can we also show that this works not only in my lab, but in other people's labs and against these high consequence pathogens? And with the luck of finding molecules like this, we have found such molecules. So we do have small molecules that block virus replication. One of them was featured as one of the top 100 discoveries of 2013 because we found a small molecule that blocks not only Ebola but other viruses, suggesting that we can have one drug that could actually act against multiple different viruses, which would be lovely. I don't want to belabor that. I want to talk about another thing that the lab has been trying to do, which is to try to make better diagnostics. And the reason for doing that is, I think, apparent in the current outbreak, where there are delays of up to 4 or 5 days for patients to be determined either Ebola positive or negative. At that time, when they're waiting for the response, they're in a large group of people that are suspected. As you can imagine, there are some people that are going to be positive and some people that are going to be negative in those populations. But they're all sitting in the same room. And if you can't get diagnosis done, some of the people that probably were not infected at the beginning of that test cycle will be infected by the time they get to the end. And so that's one of the things that we would like to try to block and keep from happening. And we're trying to do that by developing essentially lighter and cheaper and less energy requiring machines and less energy requiring tests. And it all goes to the central clinical problem, which is when people have a viral disease, often they show up looking generally sick. They'll show up with general symptoms like, look, I feel I have the flu, and that could mean you could have any number of infections. So how do you limit that? It means you have to do a molecular test to actually find the virus and say which one it is, and you can't just rely on somebody's symptoms. But those are often very complicated. The current tests use enzymes, and enzymes are great if you live in the West and you have a constant supply of power. You have FedEx to deliver enzymes on dry ice where you can use them. They're much more difficult to use in Africa where you have none of those. One of the other issues is a lot of tests will look for one virus. You guys may have gotten a rapid flu test or a rapid respiratory syncytial virus test that checks for one thing. When you have non-specific symptoms, you'd like to have a test that looks for about 7 different things in order to identify what you actually have. And so both of these are real difficulties for current virus detection schemes, which center based on a polymerase chain reaction assay, an ELISA assay. And also you can do direct culture to watch the virus grow out in a lab. And in terms of time, doing culture is really not very satisfactory. If it's going to take days to figure out what you have, that's not a good sign. But PCR and ELISA can be very good. They can work in a few hours. A problem is they require a great deal of sample preparation in order to use them. And they're anchored to a clinical lab. This is an example of that. So this is Andrea Marzi, a friend of mine. This is her in Liberia. She is running some of the Ebola diagnostic tests. You can see the machine there. You can see another technician back here doing the same thing. This is great work. It's really important work. However, one of the things that is limiting how fast they can do the tests is this right here. That's a power plug that's totally filled up. In order to run all these machines, they need a dramatic amount of power. This is generator based. This is not in order to put up this diagnostic facility, they needed a tremendous amount of infrastructure. It's very hard to push out of a large city and at the site of need, which may be rural. And so this is one of the things that my lab has been trying to fix, to try to make a machine that really does not need a tremendous amount of power. And the inspiration for this comes, of course, from late night TV, in my case. So this is a Boston University alumnus modeling what I hope to have as a virus detector, which is a tricorder. And what I learned from watching Star Trek is that this thing can do virus diagnostic in about 30 seconds. This is great. I want this. Also, you can see it's tremendously portable. So, of course, that's in the future. My question is, how do we get to the future? The first answer is you just wait a while and then it's here, but that doesn't work too well in the laboratory. And so one of the things that we've been focusing on is starting with a diagnostic approach that we know works and making it faster. making it easier, and making it more sensitive. So we started with an assay that's called, a platform that's called enzyme-linked immunosorbent assay, or an ELISA. And the way that works is you take your sample that you think has virus in it, in this case the virus is green, and you put on a plastic plate an antibody that will capture that virus. And when you incubate these guys together, this antibody will capture that virus. Then you need to figure out that that's happened. And in order to do that, you need to add a second antibody that has a label on it that'll shine and say, all right, I'm here. This is a great assay. These are used clinically all the time. But they take a while. One of the things that we were concerned about was maybe we could shortcut this. Maybe we could make it a faster assay by getting rid of this label step and then just go here and look for the answer. And so how do you do that? The idea that we've been following is usually in an ELISA assay, the thing that you stick the antibody to is inert. What would happen if we made it so that it was active on its own? So that if there was no virus present, you wouldn't see any signal from it. But if there was virus present, what the antibody was bound to would tell you that something was there. And that's a great idea. There's a lot of very expensive ways to make that happen. But we, of course, were looking for something that you could take to the point of care, which means it probably can't be expensive. And so we asked if we could use something really simple, like light. Light's really easy to get. It's really easy to get in all countries. And so what we've been studying is something called interferometric reflectance. What that is really similar to what happens when you see a rainbow in a soap bubble. So what is happening when you see that rainbow is white light is coming in to that soap bubble. Some of it's bouncing off the top, some of it's bouncing off the bottom. And the wavelength of light that you see represents the distance between this outer film and the inner film. What that means in terms of measurement is you are able to see 3 or 4 nanometers of difference in the thickness of that soap bubble. So your eyes are becoming phenomenally sensitive to very, very small changes. And that's really cool. It's fun to watch soap bubbles. It's fun to watch oil slicks in water. But it's also something that if you are an engineer, you can reduce to a simple instrument. You can reduce to a silicon and glass interface that will do essentially the same thing, that if you have small changes at the surface of that glass that's on the top of this little wafer here, you can measure that. You can see very, very small changes. And this was something that was being developed by a colleague of mine, Salim Unlu. This is Salim here, working the machine. This is me, looking like a doofus, watching him use the machine. And what I'm doing here is saying, can we use this to detect viruses? And the idea that we followed was this. We were going to put an antibody on this active surface, and we were going to look at it. It's going to give us some signal that comes out. And the idea is if a virus got bound right at that surface, that when we bounce light off it, now that signal would be slightly different, that there'd be a difference that we can actually quantify. The nice aspect of this is really cheap. Silicon is really easy. Putting a glass surface on it is even easier. The semiconductor industry has been helping the telecommunications industry, helping the computer industry make these types of chips for decades. We can call up California and say, we would like 14,000 chips, please, and it costs us 3 weeks of time and $1,000, which is less than $1 per chip, which is great. That's what you want an assay to cost in the end. So, and in general, the idea here is that one of the other advantages is in an ELISA, you end up using a single antibody to get a single answer. If we use this chip, we can array a number of different antibodies down on a surface. And so you can imagine we can look for multiple different things at the same time. that gives rise to the idea of this multiplex concept where we've got a whole bunch of strips of antibodies here, and you can imagine them binding various different infectious agents and reporting that out when they're present. This is something that Salim and I have been working on for the last several years, and the nice part is that we know it works now. So we can detect virus binding very easily and very sensitively, which is important for these assays. And we can look for several viruses at once. And one of the additional things is that the simplicity of the assay is really, I think, a strength of how we would like it to work. This is already published, and it's got lots of scientific mumbo jumbo in this article, so I won't belabor the point. What I do want to say is what we're at now. So the current assay looks like this. You can take a sample, which is either blood or serum, it doesn't matter. You incubate it with a little centimeter size chip for about 30 minutes, maybe 45. You then wash it and you can read it. It's a very simple, straightforward assay. If we were going to do an ELISA, you would start with the same sample, but you would only have a single thing that you could look at rather than 3, which is what we've got up here. You'd have to do that same incubation step, wash, label, wait an hour, wash again, and then read. So essentially what we've done is cut the time of the assay down while keeping the sensitivity, while keeping all of the other aspects that are good about an ELISA. This, of course, has gotten lots of people excited. We got people calling from computer world, which never talks to me, asking how we were going to use nanotech to solve the world's problems. And the reason I didn't put the article in here is what I told them is we're not going to solve the world's problems, but we might help us diagnose disease earlier. And of course, this leads to people doing additional questions of how do you address this problem, and how do we address this lack of a mobile test? Right now, the diagnostic that we've developed has been picked up by 2 separate high containment labs that are testing it. We just sent our initial chips to Sierra Leone a couple weeks ago to see how they work, and we're optimistic that we can push this paradigm so that you can get very simple testing, that you can get effective testing using this technique. And that said, I don't want to leave you with the impression that we are all the way to where we want to go. The things that we still have to do is we need to shrink our detector into a portable system. We like this size, and we'd like to get to this size. We also would really like to get to the point where the diagnostic interface that we use is incredibly simple, where you're either unhappy at your diagnosis or you're happy at your diagnosis, and we still need to work that part out. So that I've talked for plenty long. Now I think I'll stop at this point. The thing I wanted to do before I do that is to say I've stood up here and talked to you about all the fun things that the lab does. And these days, I don't do any of them. That's done by the people that you see here, and it's a large group of people. As I mentioned, a lot of the virus detector work we've done with my colleague Salim Unlu and a tremendous group of graduate students and postdocs. I'm fortunate to have wonderful people working for me. The antiviral compound that I mentioned briefly, we developed with John Snyder at Boston University. And we're also very lucky to have good collaborators outside of the Boston area, including people at the University of Texas Medical Branch, which is in Galveston. We've gotten lots of reagents from people who've been very nice when I've called them up and asked for them. They've given us antibodies when we asked. This includes all of these people here who've given us tremendous support. Out of the work that I talked to you about today, we managed to kick one postdoc out of the lab. The successful part of that was that he went and started his own company. It's called Next Gen Arrays. And they've picked up on the technology that's been developed and have now commercialized it. You can see this is the detector that I showed you in cartoon. This is David. This is his colleague, George Daboul, both of whom have been instrumental in sort of pushing this technology to where it is today and making what is now a clinical laboratory type instrument and moving towards a portable instrument in the future. So I think I will stop there. Thank you for your attention.
stops that process
Yeah.
in your body?
Right. So how do you ever win?
Die of a cold, right?
Correct. Yeah. And so the simple answer is that there are two things. The first is that there are what are called innate defenses that your cells have. So they deploy several danger sensors, both on the outside of their cells and the inside, that look for patterns associated with virus infection. And when those happen, that starts a transcriptional program that produces a large number of antiviral proteins that not only try to attack the virus that's in the cell, but also send signals to neighboring cells saying, prepare yourself, there's a virus in the neighborhood. And that can be somewhat effective. There is another thing, however, and that is your adaptive immune system. So those are your T cells and your B cells, the ability to make antibodies and that are specifically directed against whatever pathogen is in your body at that moment, and also to develop T cells that also recognize cells that are infected and engulf them and get rid of them. So there are two separate ways that the body, I've just explained two separate ways. There are probably ones that haven't been really well defined right now. But I think the second part of your question is, so why do you just get sick with some and why do you get really sick with others? That's where I think there's a big gap in knowledge right now. Right? When you look at viruses, it's not that easy to say just from looking at their genetic material, oh yes, this is going to be a bad one. No, this one won't. It's something that has to be done in experimental, and unfortunately that experimental is us.
So in the bad ones, what happens to these systems that help?
Right.
I mean, what's going
Right.
on?
So,
I guess that's what
so you can probably guess in that they don't work effectively. And one of the things that viruses are known to do is to essentially deploy their own counter defenses, right? So what I've told you is that cells have all these detectors that are ready to recognize a virus infection and send signals. And viruses, this is anthropomorphizing, but bear with me, viruses are no dummies. If they have evolved to become successful, they have figured out ways to antagonize a lot of these signals, right? So many viruses bring in molecules that they don't need in order to just complete their replication cycle. You can get rid of them and put them in cells in a tissue culture dish and they are just fine. But then you try and put them in an animal and they no longer cause disease. And when you look at what happened, what that protein does normally, you find that that protein goes in and turns off part of the immune response. Sometimes it's that innate response, sometimes it's the adaptive response. But a lot of the viruses that are associated with very severe pathogenesis make these innate response or adaptive response interfering proteins. And that's certainly part of it. But while I say that, there's also a lot of viruses that don't cause severe disease in us, that produce similar proteins. So it's not, I don't know that it's really well understood exactly what that switch is.
So the more severe ones are the smarter ones.
I would, that's what I'm going to say, while I still, at the same time, I cringe at that, at the fact that that ascribes a brain to something that has 12 proteins.
I have two questions, please. I was under the impression that it was difficult for a virus to cross from one species to another. Do we know the process? That is question one. Question two is, fortunately, the people who had Ebola and were treated in the United States are virus-free. Does that mean that they are cured?
So I'll take the second question first, and the answer is yes. So there are, so the second question was, if you have had Ebola and now you have recovered, are you cured? Are you free? Are you completely free of the virus? And so the answer that we know is yes. So the entire process of the disease is when you are initially infected, the virus, for lack of a better analogy, starts out winning. Right, so it gets into your cells, it begins to replicate, it will move to various different organs and continue to do that and make more and more and more of itself. The entire time what your body is trying to do is to dial in the right response, to dial in the correct antibodies that are going to recognize that virus, keep it from entering additional cells, and allow your adaptive immune system cells to pull each of those viruses in and sequentially destroy it. And the people that succumb to this disease do not get to that solution, right? So if they're doing a Rubik's Cube, they don't solve it before the virus replicates too much. Those that do make a very strong antibody response that recognizes the virus and parts of the virus. And what that does is it completely clears the virus from the person's system, but also will protect them in the future if they see that again. So if they have survived, they are essentially vaccinated. It's not a vaccination process that any of us would want to go through. But it is one that confers protection. So the second question, which you asked first, is you were under the impression that viruses have a hard time jumping. And unfortunately, the answer there is very specific to an individual virus. So the classic example of something that has a hard time jumping from species to species was smallpox, right? So smallpox had one organism that it could infect, and it was us. And for many centuries, it did a great job of doing that. When a vaccine was developed and disseminated in the proper fashion, and this was in the 60s and 70s that it was really pushed very hard. Once you got to the point where you vaccinated in the areas where smallpox was present, it died out. It's now no longer in circulation. There's nowhere where it's hiding, except for certain freezers at the CDC, but it's safe there. So that's an example where the virus did not have another reservoir to sit in. However, things like flu are the opposite of that. So you'll remember the 2009 flu pandemic. That virus was a virus that likely circulated in pigs, and also there's genetic evidence that it circulated in birds as well. And it was part of those jumps that allowed it to come back into humans. So different viruses have different abilities to do that. I guess another good example is HIV, which is, again, restricted to us. It does not cause disease, even in very genetically closely related non-human primates. So there are a lot of vagaries in my answer, which I'm sorry about. Viruses don't conform to a specific paradigm, but some are very easily transmitted among species, some not at all.
Question about mutation of the Ebola virus.
Yeah.
Is there any indication that it's mutated in any way since its outbreak after 1976, making it more proficient to get into cells?
Right. So the question is, has Ebola virus mutated since 1976? Absolutely, it has. Every time there has been an outbreak, the sequence has been different than the previous outbreak. There has not yet been a situation where the virus with the exact same genetic sequence has shown up. That said, a lot of the outbreaks, the viruses are very, very similar. So the current ongoing outbreak is more than 97% identical to what was seen in the Congo in 1976. So the answer is there are certainly mutations. And even now, as this virus is passed from person to person in Western Africa, there will be additional mutations. Whether that makes it easier for the virus to transmit and makes it more pathogenic, or makes it less pathogenic? I think I don't know the answer. There are two opposing possibilities. One is that as it essentially goes through life cycles in different humans, it may become easier to transmit and therefore more pathogenic, right? That's the scary one. The less scary one is as it goes through multiple cycles, it may become less pathogenic and therefore easier for the virus to transmit. So this would be an analogy to Epstein-Barr virus. Most of us have Epstein-Barr. Most of us have not noticed that we have the virus. So the virus is incredibly successful at being carried around by all of us, at being transmitted to multiple generations of people. It's not at all pathogenic. Sorry, I mistake. It's not tremendously pathogenic. It can lead to some pathogenesis, but most people really won't notice it at all. And so there are those two potentials in every virus, I would say. The potential to remain highly pathogenic and the potential to become less pathogenic. I would say from what I have seen so far, the very significant pathogenesis associated with this outbreak, I don't believe is due to the virus itself. I believe it's due to the situation in the countries where the outbreak is so rampant that is what's allowing it to be transmitted for so long. And the reason I say that is that in Senegal and in Nigeria, where they had transplanted cases, they were able to contain it very readily. And if there were a change in the transmission pattern, I wouldn't expect that.